Also, just to make sure, can you guys see my mouse? Yes, no, maybe. All right, so I'm just gonna, okay. Okay, that was not. All right. So, yeah, I know my mouse is kind of small. So we're gonna start off today, and what we're gonna be talking about today is ballistics and chromatography. So, yeah, this is your title slide, and if any one of y'all knows Elmer Fudd from like Bugs Bunny. All right, so does anybody know what ballistics is? Or like, what does ballistics have to do with? All right, so I feel like if you could, using context clues, wait, so this guy is holding a gun, right? So kind of like, okay, missiles is maybe reaching a little bit, but basically it's like firearm identification, you like gunshots and stuff like that. All right, so what does a firearms examiner do? So this is a person who mainly deals with ballistics, right? So basically you can't really determine exactly who fired the weapon because based on ballistics only. So like, yes, there are other factors that you can use, but if you're going to go on only ballistics, so rule out fingerprints and all of that, then you can't say who fired the weapon. But with ballistics, you can find out a lot of other stuff. So but like if you can explain the features of the weapon and like some other stuff. So what can ballistics explain? Because if you can't figure out who shot the gun, what can you do? So you can figure out what the caliber of the bullet is. So what the caliber of the bullet is you guys know what the barrel of a gun is. It's like it's like kind of like the place where the gun goes. Oh, so yeah, I don't know if you guys can see the Q&A. Just let me know if you can. But question was, what is forensics for, like crime? Pretty much, yeah, it's a study of crime. Yeah. So the barrel, the caliber of the bullet just means the diameter of the bullet, kind of how big it is. So you can figure out that. So using that, you can kind of find stuff about the gun. If a firearm was recently fired, their type of firearm, where the shooter was, number of bullets fired, and other stuff like about the crime. So you can also figure out this. Once you figure out what the gun is, you can also find the like safety features of this gun, and this safety feature can tell you like stuff about like how something happened. So if it was a suicide, murder, etc. All right, so here are the types of guns and firearms. So first we have pistols and handguns, and these are, fired from one hand. They're smaller than 16 inches, so the barrel length is less than 16 inches. The bullets in these will bounce around inside the body. Don't think too hard on that. You might, it's probably going to feel a little bit weird if you really think about that. And then these include revolvers and semi-automatics. Okay, and then next there are rifles, and these of course are supposed to be fired from two hands. These use shoulder support like pistols and handguns, they don't use shoulder support. These have a rifled barrel. We'll get onto that in a little bit. The barrel length is more than 16 inches, so unlike pistols and handguns, and these include bolt actions and semi-automatics, and again, we're going to get onto that later. And then there's shotguns. I wrote shotguns, just pretend that says shotguns. These are fired from two hands, use shoulder support, has a smooth barrel, and these are pump action and semi-automatics. So some examples of pistols are like Glocks, rifles, like assault rifles and stuff like AR-15 or something. Shotguns, like KS-23. You guys know what that is? Okay, so start with revolvers. So like from the name, you can kind of tell what it does, and like it has this chamber thing, not this thing where it has the bullets in, and it kind of it's like a revolving cylinder, right? So revolvers use like really powerful ammunition compared to semi-automatics. I guess that's kind of helpful. All right, now we have semi-automatics. So why they're called semi-automatics is that the like these do load automatically, but it requires trigger. So it's not like a completely automatic because it's one bullet per trigger pull. There are like shotguns, rifles, and pistols. All right, so next we have okay, sorry. Bolt actions. So these are like hunting rifles, sniper rifles, and what these do are they deliver like a single lethal shot from a distance. And what's kind of important about these are that it ejects the shell casings. So basically the shell casings kind of go away, like, if that makes sense. We'll also get onto that later. All right, next there's pump actions. These are used in law enforcement, commonly known as riot guns. I don't really know how important these are. Okay, so how does a gun work? First, the trigger is pulled, obviously, and this causes the firing pin to strike the primer. So what the primer is this little thing in the bottom of a bullet. It's showed over here, and this causes a spark. And as you can see, the bullet also contains the gunpowder, and this will ignite the gunpowder and cause it to explode. So the explosion of this gunpowder will cause up a buildup of gas. How much does a gun cost? I do not know. Can Google that. It's not very important per se. Like this will burst, kind of like think of if you put put Mentos in like Coke and then close the cap and it kind of explodes. So that shoots the bullet. And this is just for semi-automatic guns. The bullet casing is actually ejected, so it's around like up here with the casing. All right, so evidence that you can find from a firearm. These include bullet fragments, bullet casings, ammunition, and these are often viewed under a microscope. So when you shoot a gun, it leaves these little microscopic marks on the bullet, and you can use a microscope to view these. And it can kind of be thought of as a ballistic fingerprint. So these would be like kind of unique to it, because if you guys were, I think some of you guys were here, I don't know if all of y'all, where we talked about fingerprints, and fingerprints are unique to a person, so it's a pretty useful identification method. All right, so types of bullets. First, regarding to the chat, what do you mean by the big gun? The big gun, you can Google that. I will not be answering for that. Types of bullets. So you have the pistol, rifle, and the shotguns. And these all have very different bullets. So as you can see, the pistol is like shorter and like rounder at the tip, while the rifle is long and sharp. And the shotgun has a plastic casing. So as you can see, it's like red, and it has a cylinder shape, so it's like flatter. All right, so rifling. Each gun has a set of unique markings inside the barrel. So over here, you can see that there's like kind of grooves in the barrel, And when the bullet goes down this barrel, it'll spin really fast. And basically it spins I think William or something. Can you elaborate on that? Okay, so when the bullet goes, it's to host and panelists, never mind. When the bullet goes down the barrel, these will call it to spin. And what it does when it spins is this improves the aim. And it also like, it both improves, yeah, I think it's just improves aiming. And this has something to do with torque and like physics and yeah. So these will help identify the gun because usually inside the barrel, the rifling is also unique to the gun. All right, so mushrooming. After a bullet is fired, sometimes it hits like a really hard target, right? So it'll make this kind of mushroomy, like it'll make the bullet kind of, I would say, split at the top. And some will also disintegrate, but this will this is kind of makes things challenging since like you can't see the rifling as well and stuff, but as you can see, like over here in this one, you can still see some of it, so I guess it is somewhat helpful. All right, so while bullet analysis can be helpful, there are many cons. Like, so the human eye is flawed, so rifling you can't really be exact. And then the striations, that is also spelled wrong, but the striations, which are like these things on the bullet, they can eventually wear out, and things like grit, rust, and stuff can kind of give a false positive. All right. Gunpowder, this kind of deals with chemistry in a way. So if you go really deep into this, this is a lot of chemistry, but I think today we're just gonna cover the really basics. And these are not all the like same. If I remember, okay, they're not like, these are not all the components in gunpowder. And I just got a question in chat. It says, can a gun holder use the wrong bullet for a different gun? From what I know, I'm not exactly sure on this, so, but I don't think so. Like it just won't, I don't think it'll fire or something like that, or like it just won't work properly because of the different mechanics in each like gun. But so there's nitroglycerin. Well, I guess, yeah, you guys are probably better than me at this, but gunpowder is usually made like with some components. So nitroglycerin is the actual explosive stuff. So this is what combusts. And then there's diphenylamine, I don't know, diphenylamine. This is a stabilizer. It bonds with the products of the explosives. So this is another one explosive is nitrocellulose, but over here just is nitroglycerin. This will slow down the decomposition, so it'll kind of just stabilize stuff. And then dimethylphthalate. So this it will improve the flexibility and plasticity of the propellants. So in this case, like the bullet, and basically it prevents the granules of gunpowder from shattering. And next, there's ethyl centralite and methyl centralite. So these are kind of like, they can be used for the same thing. I would say like one, they're used in different guns. So like it'll be one or the other. These moderate the burning, and some so for smokeless gunpowder, because there's like regular gunpowder, it's smokeless, right? These moderate the burning and usually also act like a stabilizer. And then there is 2 NDPA and 4 NDPA, and like ethyl centralite and methyl centralite, they can also be like one or the other is found. What this does is it eliminates acidic nitrates and nitric oxide. So it kind of also is a stabler, and what I mean by stable is it kind of controls the explosion. All right, so we have some practice questions. Oh, we are going like kind of faster than I thought. So I'll say like 2 minutes per question. Or if you guys want more, you can just let me know. All right, so it's been around 2 minutes. Do you guys want more time, or do you want me to just like give the answer? All right, so since nobody else is like answering, I'm just gonna say yeah. So to the three of you guys who did answer, it is oh, 4. It's B. So yeah, the nitroglycerin, it explodes. And okay, so for is number 2, again, I'll give like 2 minutes on it. And then also if you want more time, just ask. All right, so if anybody wants more time, say so now, or else I'm just gonna go on. So does anybody want more time? Okay, I'm guessing not. So I saw some like the answers were quite mixed. But I think most of you guys got it right. It was B. It improves the aim and it makes the bullet spin, and it doesn't. The rifling does not make it go slower per se, so it doesn't really regulate the speed. Okay, so number 3. Again, I'll say like 2 minutes, and if everybody stops answering like at once, um I'll like explain. All right, so if anybody wants more time, you can say so, but I'm gonna, if nobody else like says anything, I'll get on with answering. So this was kind of also mixed. It was between C and D. So it was D. So if you look back a couple slides, like okay, a lot of slides. Let me see, like here. All right. So the gunpowder, as you can see, is actually okay. Or okay, the gunpowder is actually inside the bullet and not at the back of the gun. So over here, this is how the bullet is propelled. All right, so that was the last of ballistics, and now we'll go on to chromatography. So does anybody know what chromatography is? All right, so chromatography kind of is like pen mark analysis, I guess. You could kind of say that it is part of chromatography. So what chromatography is, okay, is it's like this way of separating compounds based on the affinity for a solvent. And chromatography, it comes from chroma, color, and graphein or graphy, which means right. So technically you can kind of think of it as pen analysis. And um, yesterday we actually talked about electrophoresis. So electrophoresis is kind of like a type of chromatography. It definitely does separate the compounds and stuff. All right, so Mikhail Tsvet is this like I think Russian guy. He used it to separate chlorophyll and carotenoids. So these are just two pigments. Chlorophyll, as you guys know, photosynthesis, and carotenoids is like it's a protective pigment, and it protects the plant from the from UV light. And I guess like it's not a super big topic. So, but like if you guys are interested, yeah, those are two pigments. And for a long time, this was actually ignored, and then the Richard guy and Edward revived his, and they also separated the molecules. And I just realized I had again another spelling error, like I don't know. All right, so chromatography has, I would say, like three different parts. So there is the stationary phase. So that's the part that stays fixed. It doesn't move. This is kind of where the pigments split. Or not pigments, sorry, molecules and compounds split. The mobile phase is the part that goes up the stationary phase. So it will I guess this will make sense a little bit more when we actually get into different types of chromatography and how they are actually performed. So the mobile phase is the part that moves up the stationary phase. I guess if that doesn't really make sense, we'll talk about it later more. And then the analyte is the substance actually being analyzed. This is also called a sample. Basically, this is what stuff is being separated. What the different parts of something is going to be separated. That'll also make sense later. But so in chromatography, there are intermolecular forces, and so there's like dipole forces. So this is the attraction between positive and negative ends of the polar molecules. So okay, I wrote water, but like in the picture it shows hydrochloric acid. So as you can see, the negative chlorine and positive hydrogen are like attracted to each other. So and then there's London dispersion forces when they form dipoles and like also kind of attract. So using these, there is capillary action, and capillary action is a major part of TLC and paper chromatography. So we'll also get into that later. And basically what it means is that the liquid like in this picture, it is colored water. It will adhere to the stationary phase. So in this picture, this isn't exactly chromatography, but just to explain, you could pretend that the water is the mobile phase and the paper towel thing is the stationary phase. So if that helps explain it a little bit better, yeah, just use that as, you can see the stationary phase does not move while the mobile phase actually goes on to. And it's not actually a phase, it's more like a part, but there's that. All right, so now it's we have the types of chromatography. There are definitely more than this. So there's like gas chromatography and stuff, but today we'll be mainly talking about thin layer chromatography, paper chromatography, column chromatography, and high performance liquid chromatography. All right, so thin layer chromatography. So the stationary phase is like a piece of glass, plastic, or aluminum, and it's coated with silica gel. And silica gel is this really, really absorbent substance. And so basically it's absorbent because it has a really large surface area. So this is also used as like desiccants and stuff. So like if you ever had, I don't know, like a pack of dried food or something like that, it'll have like a little packet of silica gel at the bottom that says do not eat, or like not for human consumption. And what that does is that it absorbs the extra moisture because it has such good like absorbency properties. And then the mobile phase for thin layer chromatography is usually a mixture of solvents, and this is decided on based on your sample or analyte. All right, so this is the process of how thin layer chromatography is done. So a pencil line is drawn near the bottom of the plate. So this is called the spotting line, or in this case, like in this picture, it's called the baseline. And the analyte, so this time like a solution of the dye, is dotted onto the line. So then the plate, the plate, which is like, again, the glass plate with silica coating it, is put into a beaker with the solvent. And for this, I think this picture is a little bit of a better picture to show, but the solvent line is really like, it's not clear. So here, I'll just like draw the solvent line darker. It's right here. And this is always placed above the solvent, or else your analyte will actually kind of disperse within the solvent and it's going to make a giant mess. And the solvent will move up eventually up the plate, and the substances will separate. So as you can see in this one, there are like 4 different dyes put 4 different samples put in here, and as you can see, there's like one compound, another compound that separate. Okay, so this is the one that actually does use capillary action. There are some types that don't, and we'll also get into that later. Okay, so paper chromatography, you can actually do this at home. So this is stationary phase. The stationary phase of this is like paper, so filter paper, like coffee filters, and there's also chromatography paper made for this, but it's pretty much just coffee filter paper. And then there's the mobile phase, which is water, but like sometimes rubbing alcohol is also used. And if you guys ever want to do this at home, just get like a glass of water and do this thing over here. Basically, you would tie the piece of paper to like a pencil or something, fill a glass up with your solvent, so like water, rubbing alcohol, and place it in the water. So again, the spotting line is drawn. So this is very similar to thin layer chromatography, but it's just not as good. So dot the ink, the analyte or sample, on the spotting line, and the paper is placed into the glass. Or this time it's like a beaker, or like honestly any kind of container that's tall will work of the solvent. So the solvent again will move up the plate, and the analyte will separate into the different compounds that you want to identify or like view. Again, if you look over here, there are the different compounds. So like over here you have the yellow, and then like the blue, or like this purple color, and blue. I guess this one's not as like good over here because it's like kind of the same color. Okay, so you know, if you guys ever want to recreate this, that it's definitely possible. Okay, so next there's column chromatography. So unlike the last two, column chromatography does not actually use capillary action. And again, if you guys don't remember, capillary action is the water or solvent being able to move up the surface. So this is kind of like a reverse version of that. It actually uses gravity to move the mobile phase and the stuff down, the analyte down. And so it's kind of like an opposite way if you think of it. So the stationary phase is also silica gel, but in this time, the silica gel is actually like very densely packed into a column or like a tube or something. So in the mobile phase, again, like TLC, this is a mixture of solvents and it depends on the analyte. So basically what you're trying to, depending on what kind of sample you're going to use, the solvents have to work towards that because there's like polar and non-polar stuff. Okay, so column chromatography. So as you can see, again, the column is filled up with silica gel or alumina. And the analyte is put over that. So like the sample is put on top of that. And then the mobile phase is added on top. So in this diagram, it's actually, it's a solvent, but it's basically the mobile phase. So as you can see, they're placed through it and they'll eventually separate into like different bands of color. And what's actually really cool about column chromatography is that you can collect the different compounds and separate them. So once this is added, it'll go down as like bands. So you can put another flask under this column thing and collect the molecules. All right. HPLC. So this is high performance liquid chromatography. So it's a modified form of column chromatography. Again, the stationary phase is silica and alumina or alumina. And this is kind of like a mixture of the solvent and it depends on the analyte. So this can be used to identify compounds using standards. We'll get into the standards after I actually talk about how high performance liquid chromatography works. All right, so the process is that, again, the column is loaded with the stationary phase, just like column chromatography is, right? But as you can see, this looks a lot different than this does. So the column, this over here, is connected to a pump, all right? And the pump pumps the mobile phase, so again, the solvent into the column. But instead of just like putting the sample or analyte into the column, you actually have to inject it in here. So again, also like this also does not use capillary action, but the solvent will go through in here and it'll go down into the detector. And the detector will detect like different compounds and the data will be displayed as like peaks or an electrical signal. So there's like this over here is an example of like the peaks, right? And some of the detectors, they can be like, there's a lot of different ones. So if you ever Google this, it'll say the type of detector, but yeah. And the detector also like sends this to a computer and then you can actually view the data. All right, so here are the standards. So it's kind of like a reference sheet a little bit, and these help identify the compound. So over here, as you can see, say at A, this substance peaked, and then the standard sample, which is kind of over here, which tells you that this is compound A. So if this peaks over here and this one also peaks over here, so that means, you know, the unknown sample, this pink labeled thing is compound A. And then again, over here, it's the exact same thing. So if it peaks over here, it's blue. This is compound B. All right, but if there is like no standard sample, you won't really be able to figure out what it is, because like over here, look at like these peaks, you can't really figure out what they are using just this standard alone. Oh, and I forgot to mention, these also show retention time. So this basically means how long the sample has been in the stationary phase. So it'll peak once it reaches the detector. So over here, like TR, that's retention time. So that's kind of how long. And I know it doesn't really show here. I can see if, okay, yeah. Over here, you can see the retention time, where like 2 minutes, 4 minutes. And so where it peaks is where the retention time is labeled. All right, so retention factor. So this is not retention time. If you guys want, you can use a calculator for this. Basically, it's a ratio between, again, another spelling error. I have a lot of those in this presentation, I just realized that. But this is the ratio between how far the sample or analyte has actually traveled and how far the solvent has traveled. So okay, I'm gonna annotate again. This is how far the solvent has traveled. So as you can see, the dot is like right here. And then this is how far the analyte has traveled. And say like over here, it already tells you this is 10 centimeters and this is 7 centimeters. So what the retention factor for this specific piece is should be like 7/10, I think. Correct me if I'm wrong, for those of you who like, I don't know, but it should be 7/10, or if you actually do it, it's like 0.7. All right, so how is chromatography used? Okay, I need to clear that, but how is chromatography used in forensics? So you might be asking, like, how is this even applied in forensics and like crime, right? So this can be used to analyze stains and inks. So a lot of the time, this is for paper chromatography and thin layer chromatography. This can be used to match a document to a specific pen. So like if you have a stain or something, you can use chromatography to identify the different compounds in the pen and identify the different compounds in the ink on a document. All right, so this can also be used to examine for substances. If you guys don't know what's like substances as in drugs and alcohol within the blood. So for doing this, this is for, what was it called again? Oh yeah, this is for like high performance liquid chromatography and column chromatography. If you guys want me to elaborate on that, I can, but for now, I guess we can just go on to the practice questions. And this one, I recommend that you use a calculator, but you don't have to if you don't want to. Or like if you guys want, you can also do this on paper, but I don't like math, so personally, I would use a calculator. All right, so since this one is a little bit longer, I'll give you guys like 3 minutes on this. Also, if you guys want to see how it's calculated again, I can also go back to the slides. So if you guys want me to go back to any slides, just say so in chat. Grade level, I don't know, that's like kind of subjective, I guess. Oh, I just realized you guys can't see the question, but someone asked what grade level is this for? Mm, well, I mean, I guess any could work, definitely. Like if it's kind of for if you don't have a background in here, in forensics, so. All right, do you guys want me to go back a slide so you can see, or do you want me to like write down what the formula is for retention factor, or? Also, as for like the actual distance, you can just kind of assume. So, um, yeah, I guess because it's kind of subjective. All right, it's been like 4 minutes and nobody has answered, so I'm gonna just assume that you guys don't know. So I'll I'll explain this. So um, remember how over here it said that, okay, um, the ratio between how far the sample has gone. So it's over here, the distance traveled by the component, and um, the how far the actual mobile phase has traveled, so the solvent front. So knowing that, this would be, so for A, it has traveled around, I would say like 0.7, and I would say also like over here, the solvent front has traveled about 4.72, or just like 4.7. And so what you would do is you do 0.7 divided by 4.7. So grab my calculator. So 0.7 divided by 4.7 is like around 0.15. I would say, like, again, rounding. And then for B, this is again, as you can see, the solvent, whether the solvent front is the same because it's just all one, like one chromatogram or something like that. And this time the distance that the solvent or the actual sample has traveled is around 4.1, 4.2. So say 4.15 divided by 4.7, that would give around 0.88. So for B, the retention factor is 0.88. And then for C, it's Zero, again, the same place where the solvent front has traveled, and C, that is around, I would say like 1.8. So 1.8 divided by 4.7 is 0.3829, or like 0.38. And one thing that you can kind of do is that if you divide one by the other and it's greater than one, that means you did it wrong. Um, and it's actually the other way around. All right, so here's the next practice question. Since this is open answer, I'll say like 3 minutes. All right, so it's been around like 3 minutes. So the difference is actually um that like they kind of work the same. And HPLC is a modified form of column chromatography. So they both use a column, and they both use like a solvent and push the sample through the solvent to collect the data. All right, this is the third practice problem, also the last practice problem. So again, like 3 minutes. All right, so it's been like 4 minutes, 4 minutes. It's false because um before again, like I explained, it was oh yeah, paper chromatography does not actually use silica gel, and even though silica gel is very commonly used, it isn't the actual like it isn't the only one used because there's also alumina, cellulose, and like some other stuff. All right, so that's the end of the presentation. Uh, all right, so I think Aaron is here, so I guess I could end. I'm gonna end it here now. I guess if you guys have any questions to ask, definitely ask now, or otherwise I could just hand it off. Wait, who's Brian? Like, I don't know. What do you mean by Brian? Uh,